Can somebody please confirm you can hear me?
We can hear you.
Thank you. All right, before we begin, are there any questions? I'm not hearing anything. So let's begin. Where's my syllabus? I don't have it up. I know we're doing lab one today, but just want to get the syllabus up and show you where we are. Lab one, ubiquity of microorganisms. This lab must be turned in by 8 a.m. Friday, along with lab 00, or else you will be dropped from the class. I'll do the dropping sometime between 8 a.m. and 5 p.m. Is there anyone who's trying to add the class who needs to register? All right, I've got a note here. Oh yeah, I'm supposed to tell you guys if you're in the lab and your cell phone is ringing, the procedure will be to, and you want to answer the phone, the procedure is to take off your gloves, take off your lab coat, walk over to the cubbyhole or the cabinets where your book bag is, take the phone out of your book bag, and then leave the. Oh, wait a minute. After you take off the gloves, you have to go to the sink and wash your hands. Sorry. And then you walk over to the cabinet, take out the phone from your book bag, and then take the phone outside. Now, by this time, the phoning probably won't be ringing, but you can call them back. So to use the cell phone, you got to go outside of the room just because we don't want to have disturbance in the classroom. So take the phone outside. And then you'll probably have to call them back. Then when you're done, hang up the phone, come back into the room, put the phone back into your book bag, go walk over to the sink, which is now in the lab proper, wash your hands, go put on your lab coat and put on gloves, and then you can return to your workbench. Any question about that? So it's a hassle using your cell phone in the lab. It is possible, like in an emergency, we do have a regular landline in the lab, and you do not need to take off the gloves to use that phone because that phone stays in the lab. Um, Obviously, you could only get a call out from that phone, but you could call out. Any question about any of that? All right. Remember, you need to take the environmental quiz. It's not actually by 8 a.m. Friday. It's what is it? 11:59 p.m. That I need to correct in the syllabus, but you do need to take the environmental quiz. It looks like it's Saturday, actually. September 27th at 11:59 p.m. I think that's Saturday. Is anyone could confirm that? Wait, look here. September 25th. So Friday will be the 26th. Yeah, Saturday. All right. And Also take the plagiarism quiz by 11:59 p.m. this Saturday. You can take this quiz 7 times, but don't guess because if you use up all the 7 attempts, that's it. And if you do not score 100% on the plagiarism quiz questions, you will get 0 on the 100-point infectious disease project. Any questions about any of that? Now, there is one question you can miss on the plagiarism quiz. That's the very first question, and that's the um, doing your environmental scan for the Respondus lockdown browser. But that's the only question you can miss. And obviously, I don't want you to miss that question. It should be an easy point for you to get that question correct. Any question about any of that? All right, let's go to the week 1 lab. There's the module for one, lab 1, ubiquity of organisms. There's the worksheet for lab 1. I'm not going to open it there because I've got it on my computer and it's will display better if I take it from my computer. Yep. Right. Any questions about anything before we begin with the lab proper? In the labs, there's a little bit of reading that should be done before you do the labs. And because it's an online lab, doesn't matter when you do the reading, as long as you do it before you do the lab. But you don't have that button, right? Hmm. You should read for Lab 1 about culture media. That's in the third edition, custom edition of the textbook for Clark College. Read page 157 to 162 about culture media, and then read also page 162 to 163 about obtaining pure cultures. Any questions about that? In this lab, there's some video clips. The primary objectives for this lab is 1, know that microorganisms are almost everywhere. Describe the purpose of culture media. That's 2. 3, describe what colony abundance and colony morphology mean, meaning you must know those terms. And then 4, define the following terms. What's inoculation? Ubiquity of microorganisms. What's something that's sterile? What is isolation? What is incubation? What is in vitro, like in vitro growth? What is a culture media? And we have different types of culture media. There can be a liquid culture media that we call a broth. And then there are solid culture media, such as a agar plate or an agar slant or an agar deep. Any questions about any of that? I'll show you what the slant and the deep are in just a minute. Um, know what something is if it's fastidious, like a fastidious microbe. What is non-selective media? And what is ubiquitous mean? And what is a colony? Any questions about any of these terms? Let me see. Yeah, I got a picture there. All right. Microorganisms are organisms that are too small to be seen with the naked eye. This includes viruses and prions, even though they aren't quite alive. Viruses is debatable whether they're alive or not. The thing is, they do not have cells. They are not organisms, but it does include a microbe. And there you can see we're using the word microorganism for a virus and a prion, even though they're not cells. We also include helminths, worms in our studies of microorganisms, even though the adults are not microscopic, like the beef tapeworm is, I think it's something like 30 meters, grow up to 30 meters long. Maybe it's 30 feet, doesn't matter. It's a huge size. It's obviously not a microorganism. However, their infective stages are microscopic. So, of all the worms that infect humans, their infective stages are microscopic, and some of their stages in the life cycle are also microscopic. So, we do include uh, worms as a microorganism, and microbiologists have studied organisms that cause disease, calling them microbes. And that does include worms. Even though the adults are not microscopic, the eggs or the infective stage of the worm is microscopic. Any questions about any of that? All right. The phrase ubiquity of microorganisms refers to the concept that microorganisms are essentially everywhere. It's not entirely true. You can find them in places on the planet Earth where they are not present, but almost everywhere you can find microbes. This is a problem in microbiology because we usually want sterile media without having microbes in it. And how we do that is we make the media and then we sterilize it, like stick the media in the autoclave. In this lab, we're going to investigate the abundance and diversity of microorganisms in the common environment. We'll also learn basic lab protocols. A culture media is also called the growth medium, is a sterile laboratory preparation that contains a variety of nutrients required by microorganisms to grow. And we grow these in the lab called in vitro, in the tube or the plate. That's what in vitro means, growing it in the lab in a tube or a plate. Culture media may be liquid. We call these broths, and they're usually in a test tube because if they were in a petri plate, you could easily spill it and make a mess. So we usually put liquid media in a test tube. Any questions about any of that? We also have solid media. Usually we add agar to make the media solid. Excuse me here. But it is possible to add something else like starch or gelatin to make the media solid. But most of the time we use agar. We have agar in a test tube. We call this one a deep. That's just where you pour the agar in and let it solidify and it goes across straight. We call that an agar deep. This is an agar slant where you pour the liquid agar in and then you put the tube at a slant for the agar to cool and then it forms the slant. We also have agar in plates called an agar petri dish or agar plate. Any question about any of that? There are a variety of different media that we can use to grow bacteria. I'll tell you about that in a future lesson. But for now, we're only going to talk about general purpose media. This is a media that is non-selective in function. It's usually complex in composition, meaning it has many things in it. And it supports the growth of most non-fastidious organisms, like common microbes like E. coli and Staphylococcus epidermidis can grow in a general purpose media because it has lots of nutrients for them to grow. However, a general purpose media cannot grow a fastidious organism. A fastidious organism is one where you have to add certain nutrients to the media in order for the organism to grow, like um, Streptococcus pyogenes, the bacteria that causes strep throat and a whole bunch of other human diseases, like, let me see now, scarlet fever, that's one. Rheumatic fever, there's another. And a few other diseases, which I don't remember what they are because they're not very common because we now use antibiotics to stop strep throat when somebody gets it. And so the other diseases are not likely to happen anymore in a first world country unless the person refuses treatment. Anyways, a general purpose media cannot grow something like Streptococcus pyogenes because it's fastidious and it requires certain additives to the media in order to grow. Any question about any of that? All right. There are three general purpose media you need to know about. They're commonly used in the microbiology lab. Let's talk about the third one first, nutrient agar, or Na. All it is really is three ingredients. Ground up beef, we call beef extract, um, sodium chloride, water. And I think that's it. There might be one other ingredient in it. Tryptic soy agar, this is partially digested protein from soybeans, why it's called tryptic soy agar, or TSA for short. And then there's nutrient agar with added yeast extract. We call that NA, meaning nutrient agar with yeast extract. These are the three general purpose media that we use all the time in the microbiology lab. When you're using a tube or a petri dish, it should be appropriately labeled prior to its use. The following items um, should be included on any label. You should put your name on the dish, like Travis here. You should put the date that you started. That one is, I think, 9/10. I can't read the year, but a long time ago, maybe 2001. Inoculum, this is the media you're growing it in, so that they have TSA here, tryptic soy agar. And then the incubation temperature is optional. This is grown at 37 degrees Celsius. Everything else is required. The temperature is the only one that's optional. Any questions about any of that? On a petri dish, you should write around the edge of the plate so you don't interfere with looking at the plate. You can also write on the side of the plate. Let me see if I can show you that. So this would be the top. You never write on the top. You always write on the bottom of the plate. And you can write on the, no, let's say that way, the side bottom of the plate. Why you always write on the bottom of the plate is that's where the auger is. And if the lid gets separated from the plate, we now have the media and the organisms on the bottom, as well as the description for what it is. I once worked in a lab where there was a lady carrying a whole bunch of trays, let's say hundreds of trays, maybe 300 trays. I don't know, remember how many it was. And she was walking, and then she tripped. And then all the lids got separated from the bottoms. And that entire experiment had to be thrown away because nobody knew what the dishes contained. And then they had to repeat the experiment. Any question about any of that? All right. Inoculation is the placement or transfer of microbial specimen into a sterile culture medium. Incubation is the placement of the inoculated culture media in an appropriate physical environment for a certain amount of time to allow the microbes to grow. We usually incubate our cultures, meaning what we're incubating, in an incubator. An incubator can control the amount of oxygen, the amount of carbon dioxide available, the temperature, the humidity, and then the time. The time's dependent on when you stick it in and when you pull it out. That's a complex incubator shown right here. In our lab, we don't have any complex incubators that can control how much oxygen will be available and how much CO2 will be available. We only have simple incubators that only control the temperature. You shut the door and then the room's up, it heats up to a certain temperature, like 37 degrees Celsius. We can control the humidity. See this dish there? That pan, you put water in it, and that allows water to come into the air, causing a humid environment for the microbes to grow in, or at least the air. And that does keep the petri dishes from drying out within a few days. You put it in the incubator without any water in there. The plates can only survive a few days and then they'll dry out. If you put humid air in the incubator, putting water in the dish there, the plates can last at least a week. Sometimes they can last up to 2 weeks. Any question about why we humidify the air? The temperature are simple incubators. You just set the temperature, and then that'll be the temperature of the incubator. And then the time, that's controlled by you, by how long you allow the plate to be in the incubator to grow. Any questions about any of that? All right. All right. When you incubate a plate, you should put the agar plate in the incubator upside down. Get this out of here so you can see that. So when you're right on the bottom, and then you put the plates upside down, then you can easily read what's on the plate. But the reason for putting the plate in the incubator upside down is the auger is on the bottom. And if you were to put the plate in right side up, the moisture from the auger will come into the air, hit the bottom of the lid, and then form a droplet on the lid. And then that water droplet will drop back into the plate where your colonies are. And that will just disperse your colonies. So you'll get, instead of a plate with separate isolated colonies, you'll just get everything growing together. You may not get contamination, although you could, and that is if the water drips directly from the lid into the plate, it won't give you contamination. But if the water drips down the side of the lid and then into the plate, like when you're taking off the lid, for example, and the water goes down the side into the plate, that would contaminate the plate because at least the bottom side of the um, lid will not be sterile. Uh, Not the bottom side, the edge of the bottom side of the lid, meaning this portion of the lid. Can you guys see that? Let me stop my sharing, then I can see this. Nobody's answering my question. Can you guys see this? Yes, you can. So this portion here of the lid will not be sterile. The inside of the lid should be sterile. Any question about any of that? And the reason why that bottom portion isn't sterile is because it's exposed to the outside. I do need a little bit of help here. When I ask a question, please answer. How do I share this? There we go. I should be seeing my screen now. Anyways, why we put the plate in upside down is now the liquid coming out of the auger will condense on the top of the auger, which, because that's the plate's upside down, so that's where the liquid will be. And the auger will actually wick the drop of water throughout the auger, so there won't ever be a drop forming. And even if a drop were to form, it would drop on the bottom of the lid, which there's no growth there, so who cares? Any question about why we put the plate in upside down? All right. The visible bumps on the agar surface, each of those is called a colony. Bacterial colonies, if you can see them, have at least 1 million cells in them. Bacteria colonies tend to have a rather smoothly textured surface. There are exceptions to that, but most of them are smooth and, oh, come on. Trying to show you what I'm talking about. Ah, there we go. Well, I just lost it. There we go. All right. So there, that has a clear, distinct edge, a smooth edge, mm, distinct colony. That one, on the other hand, the edge is not distinct. You're not entirely sure where the exact colony is and where the plate agar is, and that's because this is a fungal colony. It's is not distinct, not clear. That's a fungal colony also. Bacterial colonies tend to be distinct and clear, even if it's an irregular edge. You can see that the edge is definitely there and then up there and there. Any question about any of that? Fungal colonies tend to be hairy or furry on their edge, and that's how you can distinguish between a fungal colony and a bacterial colony. Fungal colonies also tend to be bigger because they grow faster than bacteria.
I have a quick question.
Uh-huh.
So if we had a fungal colony and a bacterial colony on the same dish, would we still include both of those in the overall colony abundance count?
Yes,
Okay, thank you.
unless the question says the abundance of bacteria colonies, you wouldn't include a fungal colony, but usually it'll be just be the abundance of colonies on the dish. The question. All right. We're talking about a colony. Each colony is made up of at least 1 million cells, or you won't be able to see it. Usually the colonies have tens to hundreds of millions of cells. They all may be descendants of one cell placed there during the inoculation, and then that one cell divided into two, two into four, etc., by a process called binary fission. We'll talk about that later in the term. Yeah, I think this is a quick one, so we'll take a look at that one. Usually I don't watch the videos, but this one's real quick, so we'll watch it. I can get it to go. Got that one open already. Here.
Bacteria reproduce very simply and rapidly by doubling their contents and splitting in two. Just one bacterium, dividing every 20 minutes, could produce nearly 5,000 billion bacteria in one day.
Oops, not supposed to see that. All right, let's come back here. Why do I have that open? Yeah. Oh, I looked at the time for that, yeah. All right, let's continue with where we were. In theory, one individual cell leads to the formation of one colony, yet a colony could be formed by two cells stuck together or by a clump of cells stuck together. So when you see a colony, you can't be certain how many cells form that colony. In fact, that colony there probably was formed by a clump of cells because this colony is bigger than these two, which are clearly isolated, and bigger than these. Now, these may be smaller because they have other colonies around, so they have competition for resources. And that's why these colonies are smaller than these colonies, because there's no competition for these. But that one is bigger than these, and it's definitely got competition from this one here. So that one probably was formed by a clump of cells, and these probably by an individual cell. You can't know for certain, but that's my guess for why that one is bigger, because these are the same species that form these colonies. Any question about any of that? So a colony may be formed by one cell, but about 30% of the colonies on this plate will be formed by two cells or by many cells in a clump. Any questions about that? All right. Another point is you can only see colonies on a solid media. If you're growing cells in a liquid culture, all of the cells will mix together and you cannot see colonies. Let me show you a liquid culture if I got one. Uh, Maybe I have it in the lab worksheet. No, I don't have it here. Let me go online. Oh, there's one. That's obviously a broth culture. This one is cloudy, so there's something growing in it. That one, you can actually see the cells growing on the top, and then there's some cells that sedimented. No turbidity, meaning there's no cells here, and that's because the cells are growing on the top of the media. Any question about any of that? All right. The point is you cannot see a colony because all the cells are growing together in a broth culture. All right, when you see colonies, you can make out some different characteristics for the colonies. Often there will be visible colony morphological variations between different species of bacteria. We have different colony characteristics shown in this table here. The colony can have a different shape. Most of the colonies are circular, some are rhizoid, many are irregular, some are filamentous, some are spindle-shaped. I would never ask you to separate a colony that's rhizoid or filamentous from a circular colony, but I would ask you to be able to separate circular from any of these. And then there's the colony edge called the margin. There's an entire edge, like a circular colony will have this entire edge. There's an undulate colony, a lobate colony, and a curled colony, where the edge is irregular. I don't know how else to word it. I would never ask you to separate the colony and describe to me, is this colony undulate or lobate? But I would ask you to be able to tell me, is the colony entire or undulate, lobate, or curl? Any question about any of that? And then I wouldn't ask you, is the colony rhizoid or filamentous? But I could ask you, is the colony entire or is it rhizoid or filamentous? The elevation is the elevation of the colony. And to see that, you really need to take the lid of the plate off and look at the colony from the side. We have flat colonies, a raised colony that's sort of like a mesa, flat mountain. We have a convex colony, a pulvinate colony, that's just an elevated convex colony, and then an umbunate colony. I would never ask you, is this colony convex or pulvinate? But I could ask you, is the colony flat, raised, umbunate, or one of these two? Any question about any of that? I'm not likely to ask you about elevation because I'd show you a picture and it's very difficult to determine. the elevation of a colony from the picture. Like, let's go up here. Can anyone tell me what is the elevation of these colonies? Any of them? Can you tell me? It's not hard to,
No.
not easy to tell because we're looking at it from the top. To see the elevation, you really have to look on the edge of the colony. There's different sizes to colonies. If the size of the colony is less than a pencil tip, we call that punctiform or a pinprick, punctiform. If the colony is about the size of the tip of this pencil here, hopefully you guys can see this, we call that a small colony. If the colony size is larger than the eraser, or see if I have another pen here, the other end of a pen, there we go. Then we call that a large colony. If it's in between the size of the eraser of the pencil and the small size of the pencil, then we call it a moderate colony. Any question about any of that? The colony can be smooth or rough. Coming here, can you guys tell if that colony is smooth or rough? Anyone?
It looks smooth.
That's a smooth colony. It's also shiny, I'll tell you that. And that's a rough colony. That's a rough colony. Let me see if I can see any others. I don't see any others that are rough, so you have to look at the fungal ones for a rough colony here. That's a shiny and a smooth colony. Shiny is a different characteristic. That's the appearance. The colony can be glistening or shiny. It can also be dull. And the fungal colony there is dull. That one's actually dull too. The colony can be pigmented. That means it has a color visible, or the colony can be non-pigmented. Cream, tan, white, off-white, those are all non-pigmented colonies. So these right here are all pigmented. That's a slightly different colony than that one. But they're all yellowish, so those are pigmented. Those are the only colors I actually see are yellow. These right here, that gray, that white, these are all non-pigmented colonies. And then the colony can have an optical property. The colony can be opaque, translucent, or transparent. Can anyone tell me, is that colony here opaque, translucent, or transparent?
Opaque?
Opaque?
Opaque. Actually, I think all of the colonies on this plate are opaque. Let me look for one which is translucent. Colony A, at least on the edge, is translucent. There's another one that's translucent. There's a better one that's even translucent. This one's really translucent. Very few colonies are transparent. They look like a drop of water, a transparent colony. So only a few are transparent. Most of them are opaque on this plate. A few are translucent. Translucent means you can see partially through the colony, sort of like muddy water, slightly muddy water. You can see through the water. Let me see which one we have. A doesn't even say translucent, but that's what it is, at least on the edge. The center might not be translucent on A. It could be opaque. Yeah, we don't have any description here for that one. All right, that's the different colony characteristics. Here we see some different colonies. B has an irregular margin. And C would be an entire margin. We don't have that. Oh, there it is, entire margin there. They have the elevation, which you cannot tell. I can't really tell if that's convex or not, because you need to look at it from the side to see the elevation. But smooth, circular in shape, white colony, meaning non-pigmented and small. That might be a little larger than small, but it depends on the actual size of the colony, and the plate is probably bigger than this, but yeah, that's a little larger than small. All right, the colony abundance is the total number of colonies on an agar plate. There's too many here for me to count, so let's go up here. What's the colony abundance of this region right here? Can anyone tell me?
I'd
We're
say
skipping that
probably
one.
like 3.
No. 1, 2, 3, 4,
Oh,
5,
sorry. I was
6,
thinking
8,
diversity, not
9,
abundance.
10, 11, 12. Yeah, there's 12 colony abundance in this region right here, meaning I counted them, and there's 12 colony abundance. Now, for the diversity, that's the number of different species in this region, and I would say there's only 2. All of these are yellow ones are one species, and then that's a separate species. Why did you say there were 3 colony diversity in this number? You
I
these being
on
bigger?
my Yeah, on my screen one of them looked like a slightly different color. But it's probably just my eyes.
I don't notice a different color. There's different shapes, but shape, like I said, can be dependent on how many cells began the colony, as well as the fact that these are isolated, so they're going to be larger than these. I don't see a different color, but you can let me know if you do see a different color. Obviously, that is different right there. And I think that's one colony, but it's possible that that's 2 colonies where that's a colony growing on top of another colony, meaning that could be one of these growing in the center of that, but it's hard to say. I think that's one colony. Right there, that one I would say is at least 3. In this area, the diversity there, that's a separate colony, that's a separate colony, different species, and then that's one there. There might even be 4 there if I look closely. Let's blow this way up. See right there, that might be a little white colony, right, growing inside that colony there, in which case that would be 4 colonies for the diversity. Now, when I ask you for the colony abundance number and the colony diversity, you can be close. Like if you were to say 11 or 13, I will allow that because I counted 12. However, if you're way off and you say something like 3 or 4, I'm going to say that is wrong. For the diversity, if someone were to say 3 here and I would say 2, I'd say, well, that's kind of close. Maybe they're counting that one separate or that one separate or that one separate. And I see 2 different species there, but as long as you're close, I will allow it. But on the other hand, if you were to say something like the diversity here is 10 or the diversity is 1, I would say that is wrong. Any question about any of that? And obviously, I'm going to ask you for the entire plate. I'm separating it out to make it a little easier, like we did the colony abundance and diversity of this region here. And that's just because it's really difficult to count the colonies right here and right there, as well as right there. So I'm not going to try to count them. And I skipped this plate because there's just too many colonies on the plate to count. And the diversity is an awful lot too. That's definitely a different species than that and that. These 2 are probably different species. That's a different species. That might be the same species as that one. Let me blow that up a little bit. And then in the colony appearance, see, these 2 colonies look different. And then that's obviously different because they're different pigmentation. I don't know if these are the same as that, but they may be different. That's a little different pigmented color. That's a different pigmented color. So that's how many different species we have. 1, 2, 3, 4, 5, 6, 7, 8. Probably that's 9. That's probably a different colony than those. So there's at least a diversity of 9 here, maybe many more. I'm not sure. There's just too many and too many colonies. So the diversity is the number of different kinds of colonies on the agar plate. And you have to use your colony characteristics to identify the differences between colony types. Obviously, something like that and these are different sizes, so you can call those different colonies. But something like these two and even that one might be the same species as these. So it's difficult on size. You have to adjust the size. Here's a real world example. What is the abundance of cats in this picture?
7?
Abundance? 7, yes. 1, 2, 3, 4, 5, 6, 7. And what's the diversity of cat species in this picture?
It would be 2, right?
Yeah, there's 2 different species, lions and cheetahs. At least I think those are cheetahs. All right, there's the terms for this chapter, or this lesson, sorry. There's a neat picture of somebody's hand. You can skip the references, and I think we're on to the questions. Oh, there's the answers to the questions there. All right, everything we do in this lab will be online, so we're going to provide you with the question and the pictures to the answers. In lab module 1, we're going to look at the ubiquity of microorganisms. In procedure 1, we're going to ask the question, what is in the air you breathe? You don't need to answer that question, but here's a nutrient agar plate that was exposed to the air and then incubated to 24 to 48 hours in a 30 degrees Celsius incubator. And the first question is, what is the colony abundance on the plate? You have to count the colonies. Yeah, let me blow this up a little bit. Oh, actually, that's pretty good. I can get it a little larger. I got that. Ah, shoot. I downloaded, what is it, Grammarly, and it puts this on the web page, and I don't need it for the lab, but there it is. So count the numbers here. Be careful, you have to count these 2 little ones. There's a little one there. Here it's difficult to count, but that's probably 1, 2, 3, 4, 5, and then 6 being that colony there. And then there might be some colonies there. I won't necessarily count that, but this right here will be 1, 2, 3, 4, 5, 6, 7, at least 7. So do your colony abundance, then give me what's the colony diversity on the plate. Like I said, you need to be close, you do not need to be exact. And then 1C, procedure 1C, what step would you use to take to reduce airborne contamination while you are inoculating the plate? So here we got a plate. Hopefully you guys can see this. Let me take these out. And we're inoculating the plate. What steps can you take to reduce contamination while you're inoculating the plate. So don't give me an answer. Something you can do before you inoculate the plate or something you can do after you inoculate the plate. Any question about that? All right. Procedure 2, what gets on your arms when you put your arms on the lab bench? What we did was we inoculated the lab bench on a plate before we disinfected the lab bench. And there you can see the growth on the plate. And then we disinfected the lab bench. And then we smeared the lab bench and put it on the plate. And there you can see the plate after disinfection. Not the plate, you can see the colonies on the plate of the lab bench after disinfection. The first question is procedure 2A, did table disinfection work? And then procedure 2B, what is the purpose of allowing the table disinfectant to air dry? Or why do we air dry the disinfectant? So when we put the disinfectant on the table, we then smear it around with the paper towel. Let me get a paper towel here. So we're going to pretend this is the place we're sterilizing. We then move it around, and then there's a light remaining liquid on the of disinfectant on the area we stabilize in your lab bench. We could get another paper towel, meaning a dry paper towel, and totally make it dry. But we don't do that. We use one paper towel, smear it around, and then we let the disinfectant air dry. Why do we let it air dry? Is the question. And that's procedure 2B. This one is a thinking question, although I imagine you can probably get an answer if you were to use AI.
Is it because you want like to kill everything that's on the surface, basically? And if you're
Uh,
to just smear it?
I'm not going to give an answer here, but you were on the right track.
Oh, okay, sorry.
Okay. Procedure 3. What is on your hands? So what we do is we inoculate the Petri dish with somebody's fingers. There's a finger, there's a finger, there's a finger. I think that's the palm of the hand there, and probably there. And these fingers aren't quite as distinct, but you can see them better on the left hand. And what we did was we inoculated somebody's hands on the plate before they washed their hands, and you can see lots of growth. And then the person washed their hands and then dried their hands, and then they touched the plate. And here you can see the right hand, some growth, and there's the left hand, some growth also. But less growth. And you need to dry your hand because when I first was doing this lab many years ago, I quickly realized that my students were getting more growth after they washed their hands on the plate than when they had their dry hands and unclean hands and touched the plate. And I asked them what they were doing, and what they were doing, all the students were washing their hands and then touching the plate with their wet, clean hand. And the trouble with that is there is some bacteria remaining on your hands after washing them. And then the water on their hands was wicking the bacteria onto the plate. So they were putting more bacteria on the plate in their clean hands than they were putting on the plate with their dirty hands. So you need to have the dryness of your hands similar. And obviously, initially, your hands will be dry and dirty. And then when you clean them, you have to make your hands dry and clean. And there's very few bacteria on the paper towels. And they were said, well, we didn't want to touch the paper towel to get our hands dirty from the bacteria on the paper towel. And I said, no, you have to have the dryness of the hands similar, or else the water will wick the bacteria to your plate. So you need to have your hands dry after you clean them, and then touch the plate, and then you'll get something looking like this. The first question is, does hand washing work? And then the second question is, suggest a reason why hand washing did not completely remove all microbes. Like we have some microbes here. Why did soap and water not remove all of the microbes from your hands? You should provide a brief citation of your information source for that question, but this is a difficult question, and many of you will get this one wrong. So I'm warning you here. It's a thinking question. You can't get it if you think about it. You can probably get it if you Google AI also. And it has to do with the cleaning agent you're using. We'll word it that way. Meaning if I were to clean my hands with bleach, for example, I would have much fewer bacteria than if I were to use soap and water. Why might that be? That would be a hint for number 2. All right, procedure 4. When you kiss your significant other, what exactly are you giving him or her? Meaning what's on your lips and what's on your tongue. So we're talking about French kissing because we're talking about your tongue. This is a plate of somebody's lips, the bottom lip and the top lip. And obviously you can see lots of microbes on the plate. And here is a plate of somebody's tongue. You should note I've got a note here above the picture talking about the tongue plate. You should read that note. And then in procedure 4A, answer which plate has higher colony abundance, the lips or the tongue. And procedure 4B, why do you think that this is the case? Even though you might expect the abundance of both body sites to be the same, considering they're physically next to each other, you probably occasionally lick your lips, so the tongue bacteria will get on your lips and the lip bacteria will get on your tongue. So what might cause the observed difference in colony abundance? And I'll give you a hint here. You must name 2 of the 3 or more reasons why there's a difference in the abundance of the lips to your tongue. Meaning there's easily 3 reasons why there's more in one than the other. Meaning why there's more on one part of the body, the tongue or the lips, than the other. And one answer will not be enough. You need to provide at least 2 of 3. And like I said, I've had students come up with 5 different reasons why there's a difference in abundance, but 3 are very clear. Okay. And then number C, why was the tongue plate incubated at a higher temperature than the lip plate? I didn't mention that, but let's go up here to the procedure. When they took the lips, we grew this plate, touching the lips to the plate. We grew this plate at 30 degrees C. And when we touched the tongue to the plate, we grew this plate at 37 degrees. Why did we grow them at different temperatures? And that's a pretty easy question, actually, if you think about it. Procedure 5, are microorganisms in the soil? What we did was we grabbed a little bit of soil, put it in some water, and then took a dilution of that and a sample of that, and then put it on the plate. So this is not soil smeared on the plate. It's actually, we put the soil in some water and then made a dilution of that and then put a sample of that dilution on the plate. And then we put the plate in the incubator at 30 degrees C for 24 to 48 hours. And it says, what is the colony abundance of this plate here? And it's asking you, is it even possible to accurately answer this question, please explain your answer. And then, is there greater abundance of colonies from the soil compared to the lip plates? I'll give you a hint here. Some people look at all the small colonies on the lips and say there's more small colonies than there are large colonies here. And they say, oh, I see lots of large colonies, and there's more on the lips because of the small colonies. However, pull that up just a little bit here. We can see a large number of small colonies right there. There's a large number right there too. Large number right there. So there are small colonies here. It's just that there's so many, they grow together and you can't see the small colonies. And there are some large colonies, which they tend to be more visible because they're large enough, even though right there and right there, it's impossible to even see the large colonies. So I'm going to warn you, there's lots of small colonies in the soil. And it's just that there's so many, they're being hidden. And there are some large colonies on the lips, by the way, too. I would agree there's more large colonies in the soil, but there's lots of small colonies in the soil. So that's a hint for procedure 5B. Is there greater abundance in the colonies from the soil compared to the lips? Most students do get this answer correctly. All right, any questions about this lab? If not, I'll be here until 4 o'clock to help you with any questions you have. After 4 o'clock or the last student logs out, I'm going to log out. But I am here to help you, so if you have any questions, please ask. Let me ask one last time. Is there anyone here who wants to register for the class, but they are not currently registered? All right. Go ahead and get started in the lab. I'll be here until 4 o'clock or until the last student logs out.
Yeah. So for the procedure 5 question A, do you want us to give like as close as accurate of an answer for the colony abundance, or do you want like 200 plus as an answer?
200 plus would be sufficient. You don't need
Okay.
to even give a number. You could make the statement that it's impossible to count because there's so many. I mean, it's up to you how you answered that one.
Okay, I
There's
see.
lots of right answers. Yeah. Okay.
Of course. Thank
All right.
Yeah. Thank you. I'll see you later.
All right. All right. That's the last student. So I'm ending this.